Hi everyone, welcome back. So last time we talked about the lag, log, stationary, and death phases of bacterial growth. We also talked about concepts like doubling time and things like that. Previously, we explored how this rate of bacterial growth, the speed that they use to double themselves for bacteria to actually divide, it's pretty variable across species. So, like last time I talked about how we have different species, like this Vibrio species that, can replicate incredibly fast, can do, division in less than 10 minutes. And then other species, it usually takes, 12 to 14 days, like Mycobacterium leprae, which is responsible for leprosy. While doubling time doesn't just vary across species, it can also vary within species based on the conditions that these organisms are living in. So the same species, the same strain, can actually exhibit a huge amount of variation in how long it takes for it to double. So E. coli, for example, E. coli living in our gut, the doubling time for that is around 1.5 days when it lives inside of our intestines, which is actually a pretty good environment for it to be living in. It's a pretty nice place for E. coli anyway. But if that same E. coli is living in water bodies, just the local rivers or streams or ponds or lakes, which it can survive in, the doubling time is doubled. So instead of it being about 1.5 days, now it takes about 3 days in this less ideal environment. But if we grow this same species in the lab under laboratory conditions, doubling time is extremely fast, about 20 minutes. And that's because laboratory conditions are really about as close to ideal conditions as we can often get. So these conditions that different species can be under as they are trying to survive have a really strong influence on growth rate. Less ideal conditions can become a challenge for the bacterium to overcome. It has to divert a lot of its resources that would normally go to replicating, to making more of itself. Under these less ideal conditions, it has to use a lot of those resources towards basic survival needs, because it kind of requires more work under those conditions, and as a result, we have a slower rate of replication. Okay, more ideal conditions. On the opposite end of the spectrum, the more ideal the condition is, the more rapid the growth rate is. So, when the species has plentiful resources, when it's got, ideal environmental conditions, and especially when it has few or no competitors, these qualities really increase the rate of replication for these species. And like I mentioned before, laboratory conditions are near optimal, at least the best that we can get them. And it's hard sometimes to figure out what those optimal conditions are. They are not the same for all species. But for some species like E. coli, which is one of the most well-known bacterial species in existence, I would argue the most well-known, that we have a pretty good understanding about what it needs for its survival. So species can survive in a pretty wide range of conditions, though. It doesn't have to be optimal. As we saw with E. coli that could live in a variety of habitats, it does have an impact on the species. It can change the rate that it replicates and things like that, but it can survive there. That's the important thing. This variation of conditions within what we would call a livable range is, this is going to have a lot of variation for our doubling speeds. Exceeding that livable range, though, if you go beyond that, what we consider to be livable, that can mean death. So if you put the species in an environment that is so unideal that it has created, a challenge that is impossible for it to overcome, essentially, like an extremely hot environment where it normally doesn't tolerate those conditions. That can be a significant problem and can result in the death of the organism. Temperature is a great example of this. All bacteria are going to be able to tolerate a range of temperatures. None of them have such strict conditions where they can only tolerate like 10.7 degrees Celsius, and anything above or below that exact threshold will kill them. That's never something that we experience. But what we do see is that there is a range of temperatures that the species can survive in. One of them, a specific area of that temperature will be optimal. It will grow best under those conditions. And then if you start moving outside of that optimal range, it does less well. And if you move really far outside of that range, you move out of the livable range, now you are going to see the organism likely die. So again, these conditions, we're going to be finding an optimal temperature for different species. There will always be this sort of ideal condition that the species can survive best in. Exceeding that reduces the growth rates. If you go too far out, that's going to result in death. And different species are going to have really different temperature ranges. We actually have important terms that we use to describe species that fall within these different categories of like their specific temperature needs. So we have psychrophiles, that is the cold-loving species that thrive in -5 degrees Celsius to 20 degrees Celsius, okay? And just for point of reference, 0 degrees Celsius is where ice freezes, okay? So they can survive happily in conditions that are colder than the temperatures needed to make ice. So like these are species that they're not that they could survive under those conditions. It's that they like those conditions. They enjoy and need those conditions. Mesophiles are moderates. They're, like we are more like a mesophile. Like the conditions of our environment that we really prefer are kind of within this range. There's a lot of species that are mesophiles where they live between like 15 degrees and 45 degrees Celsius, where 45 is quite hot, and now we're looking at like over 100 degrees Fahrenheit. Okay, thermophiles are heat-loving species. They love high temperatures, 45 to 80 degrees Celsius, and hyperthermophiles are on the extreme end of this. Okay, where 70 to 105. Where 100 degrees Celsius is 212 degrees Fahrenheit. This is the boiling point of water. Okay, so hyperthermophiles are very extreme. Now, this is temperature is so influential for different species, particularly because of its relationship to the proteins that these species use to do all the different jobs they have to do over the course of their life. These species will make tons of different proteins, and proteins get their function by the shape that they take. So if they lose that shape, if the protein unfolds and becomes more like a ribbon, then that is really problematic for its function. It can totally destroy the function of that protein. This is a process we call denaturation. Denaturation just means losing the shape of that protein. Now, when temperatures increase, proteins have, just like the organisms that they belong to, a functional temperature range. They have an optimal range, like a framework of optimal temperature. That if the temperature is within this realm, then our proteins do best under those conditions. Okay. Now, like for example, our body sits at a typical temperature of around 98.6 degrees Fahrenheit, more or less. There's a lot of variation there, but we are looking at, because that's what the temperature that our body is maintained at, we're looking at this as our optimal temperature range. So our proteins, the various proteins that our body makes. Well, most of those proteins have an optimal temperature range that is right around 98.6, give or take a couple degrees here. But if you start moving too far out of that optimal range, well, the functionality of those proteins is going to decline as they start to denature. As the temperature exceeds the maximum temperature range for that species, for that protein, the proteins are going to lose their shape. They're going to undergo denaturation, and they're going to cease their original function. They'll have been destroyed. They'll no longer do the job that they used to do. And that can be extremely detrimental for the organisms. This can lead to death. And then when temperatures exceed the minimum for the species, so now we're getting too cold, well, proteins don't actually denature under these conditions when the protein gets too cold, but the rate of reaction slows. They don't really denature, but they stop being very useful because the reactions they normally would regulate become less and less and less and less common and require, more energy in order to make happen. A similar problem arises when the livable pH is exceeded, either moving too acidic or too basic, too alkaline for the bacterium to actually tolerate. Similarly, the proteins also have this tolerable range where if you go too far out in one direction or another, they can denature, they can lose their shape and therefore their function. In the case of proteins and pH, moving in either direction will be detrimental to them. Not just becoming too acidic or too alkaline, but either one can result in denaturation. Now, some species have evolved to survive in some of these conditions that we would consider extreme. Like, for example, really high temperatures, if you think about like the thermophiles and hyperthermophiles. And then we have also species that can survive in super acidic or alkaline conditions too. Those are the acidophiles or alkalophiles. Well, those species can survive there in part because they've evolved their proteins to be very tolerant to those specific extremes. So if they live in these high acid conditions, for example, well, their proteins are constructed in a way that they can tolerate these more acidic environments, that they don't denature under those conditions. And instead, if you start moving outside of those conditions, say, for example, you go towards neutrality or an alkaline pH, if these proteins are specialized for acidic conditions, then they would denature. So these organisms that live in these extreme conditions, that they thrive in these extreme conditions, these are what we call extremophiles. Species that are adapted to survive in extremes and have defenses to help them survive under those conditions. So most bacteria grow pretty optimally under relatively neutral pH ranges. Between 6.5 and 7.5 usually. These are species that we call neutrophiles, but there are still a lot of species that are acid-loving or base-loving. These are the acidophiles and alkalophiles, respectively. Okay, so there's a lot of variation here, a lot. For example, we have species that, even within our own intestines, and our own just digestive system in general, can survive under these unusual conditions and prefer them. There's a species called Helicobacter pylori that loves to live in the human stomach. I believe it's like 30% of people live with H. pylori in their stomach without it ever causing any issues. They prefer that environment. It's a medically important species, though, because it's also one of the leading causes of stomach ulcers in some people. So this is a species that is an acidophile. It loves high acid conditions. Okay, so our stomach sits at like a 1.5 to 3.5 on the pH scale. It's very acidic, very acidic, and so this is an environment that this bacterium actually prefers. But a lot of other species would not be able to survive under the conditions of stomach acid, and that is also what allows our stomach acid to be quite an effective barrier for potential invaders that would want to come into our body. It can be kind of a filtering mechanism to help eradicate a lot of species that can't survive those conditions. This is H. pylori, by the way. This is 40% of American stomachs. And so, peptic ulcers can be caused by this, but it can also be caused by other things too. But it's a very important organism in the sense that it is a leading cause of those peptic ulcers. Then we have salt. So high salt environments can cause another challenge for organisms that live in them. Salt's really important. It's a very important component in a lot of biochemical reactions, and it's broadly important. But if it's too abundant, particularly in the environment, that can cause some significant issues. Not because it really interferes with specific metabolic processes or something like that. It's actually a water regulation issue. The trouble comes from the processes of diffusion and osmosis. Okay, so hopefully this is review, but diffusion is a natural process where molecules that are dissolved in a liquid want to move in a particular direction. They want to move from places of high concentration to places of low concentration. So if you think about it like this, if you go to make Kool-Aid, I make Kool-Aid every now and again. Who doesn't like Kool-Aid? Well, you put the little packet of flavoring into the pitcher after you filled it with water, and what happens? Well, it's concentrated at the moment that it hits the water. But if you don't stir or anything, it will start to spread out. It'll fall to the bottom and then begin to dissolve. And as it dissolves, it spreads out and out and out. And if you leave, say that you, go to get the mail, go to walk your dog, whatever, come back and look at the pitcher after 15 minutes, it will have changed color because the dissolved material that you added there has diffused from the area of high concentration into the area of low concentration, which is the rest of the pitcher. Osmosis is a diffusion process as well, but it only occurs across semi-permeable membranes, like, for example, our cell membranes. So our cell membranes, if you remember, they don't really keep everything out. There are some things that can easily move between the phospholipid bilayer, between the phospholipids of the bilayer. Charged particles and big things like glucose molecules, they're not moving that way. Ions are not moving that way. But water molecules can squeeze through the spaces that exist between these membranes, within these membranes. And that creates an opportunity because when you have concentrations of something like salt, for example, the ions that form salt that are maybe higher in concentration on one side of the membrane and lower concentration on the other side, well, diffusion wants to do this. It wants to move the salt across the membrane from that area of high concentration to the area of low concentration. But here's the challenge. Those ions are charged, and so they can't move across the membrane. The membrane is a barrier to them. And so, in order to equalize the concentration of salt on one side of the membrane versus the other, water will move and salt will stay in place. And this is all of this, again, this is just to equalize the concentration of the thing that is dissolved in the solution, in the solute, we call it, of the solute. So osmosis, again, it's when the solvent moves, the water in this case, moves from low concentration to high concentration, trying to make those two concentrations equal on either side of the membrane. That was very long-winded, but the impact of this is important, okay? So when we talk about cells now that are being affected by this process of osmosis, there's a major impact this can have, because when you have a difference in the salt concentrations outside of the cell versus inside the cell, that can result in water moving unequally across that membrane. Ideally, we would like this. We would really like to have this right here. We would really like to have this normal in and out, where our cells, any cell that we're talking about, would have a relatively equal flow of water out of the cell as there is water flowing into the cell. But that's not always possible. If the concentrations are different, you might get the cells inflating, where water is rushing into the cell so much that it might actually pop. Or water might be rushing out of the cell so much that it shrivels up like a raisin. So these salt ions, because they can't diffuse, water is going to be moving. And when we have an environment that is very salty, a very salty environment, the water is going to be moving to the place that has the most salt. So in the case of a high salt environment, right, here's where we are. We have water that is going to be rushing out of the cell into the salty environment, trying to equalize the concentration of salt that is in the cell versus out of the cell. And the result of that is the cell's going to shrivel up and shrink, and that can be very detrimental for cell function. This is a process we call crenation, cell shriveling. On the opposite end of the spectrum, that can also be detrimental, because in a low salt environment, where the cell is saltier inside than the outside environment, then water is going to be rushing into the cell in order to equalize those concentrations. This is where you get the potential for lysis, the popping of that cell. Now, a crenated cell is very problematic, because the cell is going to be losing a lot of water and therefore a lot of function. Water isn't just important in certain metabolic reactions. It's also a facilitator. It's an environment inside of the cell. And if you lose that environment, the cell can also lose a lot of function. And in some cases, it can even die. But there are some species that actually have ability to survive under those conditions. These are the halophiles, okay, the salt lovers. We have a species that can be obligate halophiles, as in they require these high salt conditions in order to survive. And then we can also have facultative halophiles, where they can tolerate salty conditions, but they can also live without it, right? Like they like the salt, but also they'd be okay if it wasn't so salty. Species like Staphylococcus aureus is a good example of a facultative halophile. You can grow it in environments that are not salty, but it loves to live in salty environments, like our skin, for example.